Hello everyone. Today in this video, I'm going to tell you about UV Visible Spectroscopy, a must-do topic by all GATE PT aspirants. Spectroscopy derived from two words, spectrum, which means image in Latin, and scopia, which means observation in Greek. Spectroscopy is a science that the deals with the interaction between various types of radiations and matter. So in spectroscopy, basically we study that what happens when the radiations like ultraviolet, visible radiation, infrared, microwave, X-ray, when they interact with matter, that is something which is mass and occupies space. Now, question arises why this particular technique is very, very much important for all the GATE PT aspirants. So here are the reasons. It's a very important and scoring topic from exam point of view. If you go through GATE BT previous year question papers, you will find that there is definitely a question on UV visible spectroscopy. Now, the question can be either numerical answer type or multiple choice question, which usually carries 2 marks. So in numerical answer type, they'll give you the the question, and there you have to apply, in order to solve it, you have to apply a formula and you have to do the calculations and you have to find out the answer. So maybe the questions are based on determining the concentration or molar absorption coefficient, right? And when it comes to multiple choice questions, so they can give you an application based question, like they can ask you that which particular spectroscopy technique can be used to determine the alpha helix structure of proteins. And in the options, they can give you circular dichroism also, UV visible spectroscopy also, infrared also. So unless and until you are not familiar with the principle of these technique, you are not able to answer such questions. And even if you're preparing for other exams as well, like if you're preparing for GATE BT as well as for CSIR NET, then this particular topic is common for both. So this is one of the advantage of preparing this topic. So what UV visible spectroscopy is all about? It's an analytical technique. That is, it's the technique that we used for analysis of sample. So your sample can be any biomolecule, like it can be DNA, it can be RNA, it can be proteins, it can be any organic compound. So using this technique, you measure the absorption of the radiations by your sample, absorption or the transmission, like what happens when UV visible light passes through a sample. So is it how much it is absorbed or the transmitted to the sample, it can be measured. And this absorption or transmittance depends upon the sample composition. So we can say that this particular technique, it provides an information on what is in and at what concentration in the sample. Let's say your sample contains DNA. So we can find out whether the DNA is there in the sample, and if it is there, at how much concentration it is present, right? Similarly, whether a sample contains RNA or proteins, and if a RNA is present or protein is present, then at what concentration they are present. So we can say UV visible spectroscopy is a qualitative and quantitative technique. Now, since in this particular spectroscopy technique, we are using ultraviolet and visible radiation. Please remember, you might have heard about different type of spectroscopic techniques like infrared spectroscopy. You have heard about UV visible, the one we are discussing right now, right? So how they are named? They are named on the basis of what radiations have been used in the study, right? Which radiations have been used for the analysis of your sample. So here we have used ultraviolet and visible radiation, and this is the reason the name is given UV visible spectroscopy, ultraviolet visible spectroscopy. Now, every electromagnetic radiation has specific energy, frequency, or wavelength. So when we talk about the wavelength of ultraviolet region, it ranges from 10 nm to 380 nm, whereas visible region, it ranges from 380 to 780 nm. So let's find out what happens when these electromagnetic radiations interact with matter. So when the UV visible radiations, let's say the radiation which have a photon of energy h nu, it strikes the atom or a molecule, right? So the valence electrons which are present in an atom, they undergo transition from the ground state to the excited state. That is, they undergo transition from one energy state, which is lower in energy, to the higher energy state, which is higher energy state, which is the excited state. So I'm sure you all know that atom is a basic unit of matter, and atom, it has a central nucleus, and electrons, they are present around the in the orbitals around the nucleus. So there are three subatomic particles: protons, neutrons, and electrons. Protons and neutrons are in the nucleus, and electrons are present around the nucleus. So let's say this is the electron present in this orbital. Now, when the photon of light h nu, it strikes this atom, let's say it strikes this atom. So basically what happens, this electron, they undergo transition. Let's say one electron is there, so it undergo transition from one energy state to higher energy state. So here the different energy levels you can see here in this diagram, which is a Jablonski diagram. So it's a diagram that illustrates the electronic state of a molecule and the transition between them. So you can see that the transition of an electron can take place from one energy level to another energy level. Always remember, the energy states, like the one lower energy state, we are calling it as ground state, and higher energy as excited state, they are not equal in energy. So the energy difference can be given as delta E, which is the difference between the energy state of excited state and energy state of ground state. So that means this photon which is coming, which is striking to an atom or a molecule, it should have the energy, that is h nu, where h is the Planck's constant and use the frequency. The photon energy n nu should equal difference in energy, that is delta E, present between the two states. So this is very, very much important. And transition will always take place between the two electronic energy levels. So this photon of light which is absorbed, that can be detected using the UV visible spectrophotometer. Now, I would like to tell you one more point here as we are talking about the absorption. Always remember, the structures which are responsible, the molecular structures which are responsible for interaction with electromagnetic radiations are the chromophores. And these chromophores, they are present in your nucleic acid, in your protein, that is in your biomolecules and other compounds. So like for example, as you know, proteins and the nucleic acid, these biomolecules are very important. So when we talk about the proteins, the chromophores which are present in the proteins are the peptide bond, which can absorb at a range of 180 to 230 nm. That is the far UV range light. Then when you talk about the aromatic residues, like proteins are made up of amino acids, right? And if you go through the classification of amino acids, we have aliphatic amino acids, aromatic amino acids. So specifically, the aromatic residues are responsible for absorption of light. And mainly, like when we talk about aromatic residues, so they are tryptophan, tyrosine, and phenylalanine, where major contribution for the absorption of the light is being made by tryptophan and tyrosine. So the range is 260 to 280 nm, that is, they absorb 260 to 280 nm light, that is near UV light. Then prosthetic groups, that is flavin, carotenoid, heme, these are also chromophores in proteins. Coenzymes are there, FAD, so they absorb at 438 nm, NADH at 340 nm, and NAD plus at 260 nm. So from exam point of view, you have to remember the range, which is, that is, you have to remember that when it is a peptide bond, so at what range it absorbs, or if it is a coenzyme, at what wavelength range it absorbs. Then coming to nucleic acids. So nucleic acids, you know, the basic unit is nucleotide, right? Nucleic acid is a polymer. So basic unit is nucleotide, which comprises of three main components, that is the deoxyribose sugar in the DNA, and the phosphate group, and the nitrogenous bases. So sugar and phosphate really remains the same, but the difference is in nitrogenous bases. Like it can be an adenine can be attached, or guanine can be attached, or cytosine can be attached, right? Or thymine can be attached. So nitrogenous bases broadly classified into two types: purines and pyrimidines. Purines, which are adenine and guanine, and pyrimidine, that is cytosine, thymine, and uracil in case of RNA. So RNA does not have thymine, it has uracil. Now, if you go through their structures, right? So if you go through their structures, some are one ring structure, like pyrimidines are single ring structures, and purines are double ring structures, right? So because they have the aromatic ring structures, they are definitely being responsible for absorption of light. And the absorption usually occurs between 260 to 275 nm. So these are some very important points that you all have to remember. Now coming to electronic transitions. Always remember that when any atom, like how a molecule is formed, like let's say hydrogen molecule. So definitely two hydrogen atoms, they interact to form a hydrogen molecule, right? So bonding and anti-bonding orbitals are formed when atoms combine into molecules. Now electrons in the molecule, it can be classified into the sigma electron or the pi electron or non-bonding electron. So sigma electrons, they reside in sigma orbital, sigma bond molecular orbital. Like for example, when you talk about ethane, right? So this is the single bond which is present, and basically it comprises of sigma electron. So sigma electron, they reside in the sigma bond molecular orbital. Then pi electrons you will find in unsaturated compounds, right? That is those which are having the double bonds or triple bonds. Like when you talk about the ethene, right? So here in ethene, the very first bond form will be the sigma bond, and another will be the pi bond, which will be carrying the pi electrons. And then you have the non-bonding electrons also. So these are the paired non-bonding outer shell electrons, which you will find in the oxygen or nitrogen. So these are the different type of transitions which are possible, as you can see here. So non-bonding to pi anti-bonding, that is the example, like this kind of transition you will find in carbonyls. And please remember that they are energetically within the UV visible range. Bonding transition, like here in this diagram, you can see pi to pi anti-bonding transition. So this one, it is present in unsaturated compounds. So example I gave you, like in ethene, so alkenes, and they are also energetically within the UV visible range. Now, when we talk about non-bonding to sigma anti-bonding, that is, you can see here, this is the case, non-bonding to sigma anti-bonding. So the example is in like alcohols and ethers, you will find this kind of transition. So they are not energetically within the UV visible range, and they require much higher energies. That is, you need to use the radiations which have much higher energy than UV visible radiation. And then sigma to sigma anti-bonding. So these gray arrows that you see here, they actually represent those transitions which are not favorable within the UV visible range. So sigma to sigma anti-bonding, you can see so much energy differences there, right? So obviously, if the electron has to undergo transition from here to here, it requires a lot of energy, because if you remember, I told you that this delta E, right? So there's an energy difference, that is delta E, and the photon of light which is striking, the photon of light, that is the photon's energy h nu, should equal the difference in energy delta E between the two states. So the sigma to sigma anti-bonding you will find in saturated compounds, that is those having single bonds like alkanes, and again they are not energetically within the UV visible range and they require higher energy. So always remember these arrows, the black arrows, they are showing the favorable transitions, and the gray arrows, that is these two, they are showing the unfavorable transitions. That is, it will not lie within the UV visible range. Unfavorable with respect to, they will not lie within the UV visible range. Now, as we are talking about UV visible spectroscopy, kindly note that UV visible spectroscopy is an absorption spectroscopy, because the transition of electron is taking place from one energy state to another because of the absorption of light. So when we talk about absorption spectroscopy, like UV visible spectroscopy, they are based on Beer-Lambert's law. Now, what is Beer-Lambert's law? Beer-Lambert's law, according to Beer-Lambert's law, it states that amount of light absorbed by a medium at a given wavelength is proportional, please note, to the concentration of absorbing material or solute present and thickness of the absorbing layer, that is path length of light. So here in this particular diagram, you can see, let's say this I naught be the intensity of the incident light. It passes through your sample. Let's say your sample is DNA, right? Now it passes through your sample. So we very well know that when a light, it passes through a sample, several phenomena take place. Like some light will be absorbed, some will be scattered, right? Some light will be transmitted. So especially here we are talking about the absorption or transmittance, right? So obviously if more the light absorbed, less will be transmitted, right? Because your sample is absorbing more light. So definitely less light will be transmitted, right? Or vice versa, right? So if I naught is the intensity of the incident light, it passes through your sample, and this I1 is the intensity of the transmitted light. So according to this Beer-Lambert's law, basically we can plot the ratio between the absorbance and transmittance also. So when we talk about the transmittance, it is the ratio of the transmitted intensity by the incident intensity. And because, as I told you, absorbance and transmittance, they're inversely proportional. So the absorbance can be written as the incident intensity by transmitted intensity. And we always measure the absorbance at logarithmic scale. So A is equals to minus log T, where T is, I already told you, here you can see it is minus log the transmitted intensity by incident intensity. And in order to remove the minus sign, I can write it like this. That is the incident intensity by transmitted intensity. And another formula is that absorbance is equals to molar extinction coefficient, concentration, and path length. So as the Beer-Lambert's law states that when the amount of light is passing through a sample, the amount of light absorbed by the medium at a given wavelength is proportional to the concentration and path length. So this is the path length N. So this is the formula that you have to remember from exam point of view. Directly on this formula, the questions are asked in the exam. Now, how do you measure the absorbance? Definitely, you will be needing some kind of instrument for it, right? So the instrument that is used is spectrophotometer, and the components of the instrument are that you will be needing a light source, right? So for the visible region, we use a tungsten filament bulb. For the ultraviolet region, we use the deuterium bulb. Then you need a prism or a grating, right? So basically, you need a monochromator, which can be a prism or a grating. So mono means single, right? Chroma means color. So you want a light of single wavelength. So always remember the light which is coming from the bulb or which is coming from the sun, they are polychromatic. That is, they comprises light of many wavelengths. So in order to convert into light of a single wavelength, you need monochromator, which can be a prism. So I'm sure you know how the prism works. When you pass a white light, it disperses the light into 7 colors, right? Have you heard about Vibgyor? I'm sure you all have heard about Vibgyor, right? So it disperses the light into 7 colors. So the specific single wavelength light can be generated using prism or the grating. And then basically the cuvette. So always remember the cuvette is where you place your sample, right? It can be a DNA, protein, or RNA. So for visible light, glass or plastic cuvette can be used, and for UV, quartz cuvette can be used. And the detectors are the ones which will actually measure the incoming or transmitted and transmitted intensity alternatively. So here what is happening is that the incoming light beam, you can see it has been there is some mirrors and slits arrangements as well, right? So incoming light beam, it is here you can see it passes. So this is the bulb which is producing the polychromatic light. So light passes through monochromator and it is converted into light of a single wavelength, which then reaches to the slit and the mirror. So the incoming light beam is split into the two parts by the half mirror, and one beam it passes through the sample cuvette and the other passes through your control, which you also call it as a reference or a blank. And then finally the absorbance, like measured absorbance, detector will measure the absorbance, which is the difference between the two transmitted beams of light recorded. And finally, your data will be generated on the computer screen. So using the softwares, you can also plot a spectrum, that is the graph between the amount of, that is the absorbed light and the wavelength. So it's a plot between the light absorbed, intensity of light absorbed by the wavelength. Now, specifically, as we are talking about absorbance here, so it is very much important that you should know the absorbance of the radiations specifically by proteins and nucleic acids, because usually in the exam, the questions are framed around these biomolecules only. So proteins, when we, especially in UV visible spectroscopy, questions are framed around these two mainly. So proteins, always remember, I gave you a range, right? That chromophores are there in the proteins which are responsible for absorption of light, right? And if you see their structures, they have aromatic ring structures, like in case of aromatic amino acids, in case of purines and pyrimidines. So definitely they have the pi electrons there, right? So absorbance depends upon nature of electrons. So pi electrons, they absorb the light readily and undergo transitions from one energy state to another. So proteins, although they have a range where they absorb, but when you talk about the maximum, the wavelength at which maximum absorption occur, that is 280 nm, that is lambda max. That is the wavelength at which maximum absorption occurs. So that is 280 for proteins, and for nucleic acid, that is 260 nm. Now, even as I told you, use, like if you measure the absorbance, you can determine the concentration. So if you measure the absorbance under standard conditions, it was being done by the scientists, the researchers, and it is observed that if you are getting an absorbance of 1, it corresponds to 50 microgram per ml double-stranded DNA. So that means if you're getting an absorbance of 2, it indicates that your concentration is 50 into 2, that is 100 microgram per ml. So why I have multiplied with 2? Because because 2 is the absorbance we are getting. But why I have multiplied with 50? Because we already know that under standard conditions, when the experiments were performed, the absorbance of 1 corresponds to 50. So 2 absorbance of 2 means 50 into 2, 100 microgram per ml double-stranded DNA. Then similarly, the absorbance of 1 corresponds to 30 microgram per ml for single-stranded DNA and 40 microgram per ml for RNA. Now coming to absorption and intensity shifts. Always remember that the environment in which your biomolecule is present, that also plays a very important role. So like protein, is it in a polar environment or non-polar environment? So based on these environments also, the environment in which the biomolecule is present, they affect the absorption. So the absorption intensity, like you will be able to see the shifting in the wavelength at which the absorption is occurring, so it can shift to higher or lower wavelength, or similarly the absorption coefficient can be shifted, like it can increase or decrease. So here we have few terms that you have to remember from exam point of view. So I'm sure you have heard about red shift or bathochromic shift. So it means the shifting of the wavelength towards higher side. So why red shift? Because if you see white light, I told you it has 7 colors, right? So the range is like 380. I told you, like in some books you will find 100 to 400. You will find 400 to 700 nm also for visible light. So red region lies towards the longer wavelength, and violet region lies towards the lower wavelength, right? That is near to the 400, and red near to the 700. So red shift, that is towards higher wavelength. Always remember this concept. Blue shift, that is towards the lower wavelength. So initially, whatever is the wavelength, then when you because of the changes in the environment condition, it is shifted to the lower wavelength, right? Then hyperchromicity means more color, so increase in absorption. Hyperchromicity means less color, that is decrease in absorption. So here you can see the diagram, the figure that you can remember. So this is the wavelength, you can see it is increasing from left to right, and this is the molar absorption coefficient. So bathochromic shift means, like initially, let's say your sample is absorbing at 260 nm, but let's say some group gets attached to it, right? We have some groups which increase the absorption, right? So let's say some group is attached, right? Like if you compare the wavelength of the benzene ring and that too of the phenol, then definitely there is a difference in the wavelength at which they absorb, right? So let's say OH group is attached, so definitely it will cause some changes in the absorption, right? So if initially it was 260 and increased to 280, or let's say 270, so that means the shifting of wavelength towards the higher side. So what kind of shift it will be? Red shift or bathochromic shift. And if it is towards the lower wavelength, then it is hypsochromic shift or blue shift. Hypsochromic effect or blue shift. Similarly, if the molar absorption coefficient increases, that indicates hyperchromicity, and if it is decreases, then it indicates hypochromicity. So let's see what are the factors which are affecting UV visible absorption. So one is protonation and deprotonation. So this protonation and deprotonation definitely arises from changes in pH, oxidation or reduction reactions. So in that case, always remember lambda max increases. So when I say lambda max increases, the lambda max um, like definitely what kind of shift it will indicate? It will indicate bathochromic shift as well as hyperchromic shift. Then solvent polarity, if it is a less polarity, it will be a batho and hyperchromic effect. If it is a higher polarity, it's a hypso and hypochromic effect. So these are few points I'm telling you here so that you can directly memorize them. And whenever any such question is being asked on these concepts, you can answer them. Okay. So don't forget to memorize these concepts. They are very, very much important. However, details are there, which is not possible for me to cover over here in a short in this particular video, but in our regular classes definitely we covered all these topics in detail. Then orientation effect is also there. So double stranded DNA, always remember, exhibits lesser absorption than single stranded DNA. Now coming to the application part. So what do you think, where we can use UV visible spectroscopy? So yes, you know the formula: absorbance equals to mole extension coefficient, concentration, path length. So that means you can determine the concentration, that is, you can do the quantification of compounds. Then you can determine the molar absorption coefficient also. So what you need to do is simply you have to rearrange the formula. Like if you have to determine the concentration, what you can do is simply rearrange the formula where you can write absorption by mole extension coefficient and path length. So you can calculate the concentration if you know what is absorbance, if you know what is mole extension coefficient, and if you know what is the path length. So you can determine similarly mole extension coefficient also, right? Then quality control. So purity of the DNA you can check. So here you can see that if you have to determine the purity of the DNA, so always remember, we usually take the ratio, like we check the quality by taking a ratio. We measure the absorbance at 260 and 280 and we take a ratio. So always remember the ratio of a absorbance at 260 by 280. If it is around 1.8, so it indicates that is a pure DNA is there. Pure DNA means that it should not contain RNA or other contaminants like proteins. Because see, whenever you are isolating the DNA, you have to take it out from you have to remove the membrane, you have to remove the RNA, you have to remove the proteins, right? So if any of these things remains, they will be a contamination for us, right? Anything other than DNA will be a contamination. So always remember the ratio 1.8 is for pure DNA and around 2.8 approximately not 2.8 approximately 2.0 for pure RNA. Then we can also UV visible spectroscopy, like the spectrophotometers, they are also used as the detectors in other instruments also, like chromatography, liquid chromatography. Then you can also determine the melting temperature of the DNA. So just now, as I told you that, like if you have paid attention, like I have already told you that double-stranded DNA exhibit lesser absorption than single-stranded DNA. So you know the melting temperature is the temperature at which half of your double-stranded DNA is denatured, right? So that means your double-stranded DNA is converted into single-stranded DNA. So how can we know that it is converted? So basically, when you analyze, like when you do the spectroscopy analysis using UV visible spectrophotometer, single-stranded DNA will show more absorption than the double-stranded DNA. Now let's quickly see what type of questions are asked in the exam. So here you can see on your screen, this is a question that I have taken from previous year GATE PT question paper. A UV visible spectrophotometer has a minimum detectable absorbance of 0.02. The minimum concentration of a protein sample that can be measured reliably in this instrument with a cuvette of 1 centimeter path length is, this is something which you have to determine. And the mole extension coefficient of the protein is mentioned, that is 10,000 liter per mole per centimeter. Always remember, whenever such type of question is given, the strategy of solving such question is write down what all values are given to you. Okay, first read the question carefully. The next thing is note down separately what all values are given to you. Like absorbance is given 0.02, path length is given 1 centimeter. So path length is always measured in centimeter, and by default the standard condition, the standard wavelength path length is taken is 1 centimeter. So if in any question, if they don't even mention about the path length, don't forget to take it as 1 centimeter. This is one point that you always have to remember. Then mole extension coefficient is also given. What we have to determine is concentration. So to recall the formula here, you all very well know, according to Beer-Lambert's law, it is absorbance that is equals to mole extension coefficient, concentration, and path length. And please note, mole extension coefficient, it is an intrinsic property of a compound. right? Like if I'm talking about DNA, you know, it is made up of some like it involves sugar and phosphate group and nitrogenous bases. It has some structure, right? So it's an intrinsic property. So here, coming back to the question, so we have to determine the concentration. So I will rearrange this uh, formula. So absorbance by mole extension coefficient and path length, substituting the values in this formula, when you solve it, you will get 0.000002 molar. Now, always remember the another point that you have to keep in mind while solving the question is the units, right? So here you have to answer this question, you have to determine the concentration in micromolar. So don't forget to convert the molar into micromolar. So when you do it, you will get the answer to micromolar. So today in this video, I have highlighted the most important points related to UV visible spectroscopy, and make sure that you remember all these points. If you want to score, like if you want to solve the question correctly, you want to score 2 marks, which usually the numerical answer type or the multiple choice questions based on UV visible spectroscopy carries. So keep these points in mind. So we'll see you in some another video. If you have liked this video, do share and like our channel, Biotechnica channel. So thank you all for now. Thank you and take care.